Good evening. We'll start the class as soon as a few other students join. Thanks for joining. Okay. Can you all hear me? Okay. Good. Hi everyone. I'm Shankar. So this class is supposed to be interactive. We will be having problem solving sessions where we'll be discussing questions that are related to the lectures that you have seen in that particular week. So we'll have some sample questions that will be similar to how you'll be tested in the assignment problems. So what would be helpful is if you try discussing the questions that we have here so that you get an idea about the concepts in general, which will help you to solve the questions for your assignment. So uh, whether you know the right answer or not, you can still unmute and try answering the questions. Okay, right. So we'll start. So the first question is identify the method from the following statements which is not used for natural product extraction. And four options are given here. Extraction using alcohol, removal of alcohol in rotary evaporator, polymerase chain reaction, removal of moisture or water to get concentrated crude extract. So, whoever knows the answer can first raise your hand. Here, since different words or terminologies are used, I would prefer going the other way around. Whatever option is incorrect, whoever knows the right answer can first tell why a particular option is incorrect. We'll take it from there. Okay? So you can take a couple of minutes and then whoever knows the right answer can raise your hand. Okay? No, Nikhil, we don't want the right answer to be typed here. If you know the right answer, raise your hand. I'll give you a chance. You can start by explaining why other options are incorrect. That way, we will know how the other options are also related. Do not type the right answer in the text box. Okay. Okay. Since Nikhil had typed the right answer, yeah, Nikhil, you can unmute. Can you unmute? Yeah, Nikhil, can you unmute?
Yes, sir.
Yeah. Okay. So you can choose one option and you can tell why that option is incorrect.
So PCR is used for multiplying the DNA base of interest. It is not used for extraction of natural product.
Okay. So that is the correct answer. What I wanted here is we'll have to discuss all the options, why a particular option could be correct or incorrect. That is why I wanted you to justify why an option, why one of the options in the other three is not the wrong, is not the right answer. Do you want to comment on any of the other options?
Okay, sir. So alcohol is used for extraction of multiple products, uh, phenolics and flavonoids, etc. And
Why?
the
Why?
removal of alcohol.
Yeah,
Yes,
sorry. The role of alcohol.
sir. For removal of alcohol, we concentrate the extract which we have obtained.
No, no, no.
Uh,
First, we'll discuss why do you need alcohol in the case. Can we use water or any aqueous solvent instead of alcohol?
yes, sir. Yes,
But
sir.
will that
We
be
can use
sufficient? If that is the case, why do we have to use alcohol?
different. It is for extraction of particular compound. It depends on its nature. If it is aqueous soluble or it is fat soluble, lipophilic.
Huh,
Based on its solubility, we use the uh, solvent.
right. And most of the compounds that we deal with in the natural products, do you think they are hydrophilic or hydrophobic?
For volatile, they are both kinds of product in obtained from plants.
Right.
Basically, volatile oils are in oily, lipophilic in nature, and a lot of product like phenols, flavonoids, it can be extracted by aqua solvent, if I'm correct.
Okay, so there are a few things we can just correct it. So volatile compounds have this volatile nature, which means that they become gaseous and they evaporate even at room temperature that we have here. That is one thing. And compounds like phenolics and flavonoids, they tend to be more soluble in non-aqueous solution, not in aqueous solution. So let's consider the case of flavonoids. They are soluble in ethanol, methanol, and DMSO, dimethyl sulfoxide, rather than water. So this is because of their chemical nature, and that is why merely adding water is not going to help. Which is why we will use alcohol for extracting. Was that clear? Okay, good. Uh, Anyone else? Lakshya, Shobana Priya, Gabriela, Payoja. Do you want to try and tell us why the other two options, option B and option D, why they are not the right answer for this question? Lakshya, are you there? Okay, others. Shrestha, Shobana Priya, any of you would want to try? Okay, so earlier we discussed as to why we need to use alcohol, but what we want is the concentrated product. So the extract that we get from this natural product, it can be used in multiple different, uh, how do I say, modes. You can take it orally or pack it into an injectable form, which you could inject into your muscles or tissues, which means that you need the compound in its purest form and uh, in a lyophilized form, which means it has to be in powder so that you can dissolve it in the required solvent and then use it appropriately. This means that we need to remove the alcohol which it was previously dissolved. So what we are doing here is similar to precipitating it. For that, we use a tool called rotary evaporator. That is what option B is about. And option D is about removing moisture or water. This again, it depends on what is the methodology that we use. Like, let's say we don't use a solution that contains 100% ethanol. Once we remove ethanol, there will be some moisture in it that also has to be removed. So that is why we need to remove moisture or water. Whereas option C, which is polymerase chain reaction, as Nikhil mentioned, it is used to amplify a particular DNA such of our interest. So for polymerase chain reaction, what we will do is we will design primers. These are nothing but small stretches of single-stranded DNA that will bind to the 5' end and the 3' end of our DNA of interest, and it will amplify the entire region. So that is the function of PCR. Since we are discussing about natural product extraction, PCR is not related to this. We'll go to the next question. Among the following techniques, which is not a metabolic engineering approach? Option A, endogenous enzyme overexpression. Option B, genome editing. Option C, gene silencing by miRNA. Option D, growing the plants in pots. So, whoever knows the right answer can take a couple of minutes and then start by mentioning an incorrect option and telling us why that option is incorrect. Please do not type the right answer in the chat box. Yeah, does anyone want to try? So you need to talk about a particular option and why that option is incorrect for this particular question.
Sir,
Yeah, Nikhil.
genome editing is incorrect because we can edit a genome
Huh.
to produce a product of our liking, which we desire. A specific compound
Hmm.
can be produced by gene editing.
Okay. So when you say gene editing, what are all the possible uh, outcomes that would come out of genome editing? Do you have an idea? It doesn't say gene, but it talks about
Yes.
genome. So it could have multiple different possible outcomes. So do you know what are all the possible outcomes of genome editing?
So we can produce a specific protein or an enzyme which we want to induce in a particular organism.
So that is okay. It has a term. Are you mentioning referring to knock-ins?
Uh, can you repeat, sir?
So you told we can. Okay, you can repeat yourself. I will repeat my answer after that. Yeah.
Uh, I said that we can produce a new protein or enzyme by editing the genome of a particular organism. Like we can produce insulin by injecting a plasmid DNA in a bacterial cell.
Okay, so one correction that doesn't come under genome editing. In genome editing, what we mentioned, what we refer to is we have this chromosomal DNA. We are making changes to that chromosomal DNA. So using plasmid will not come under genome editing. Although that's a part of metabolic engineering, it won't come under genome engineering or genome editing. So instead, if you take that gene from that plasmid and insert bit in a particular position within the genome, that would be the right way to do. So that has a specific term that is called knocking in. Uh, that is what I wanted to mention.
Okay, sir.
Right. Yeah. So genome editing is incorrect here because it's a metabolic engineering approach. Huh. Nikhil, so do you know of any other terms or any other methods or outcomes of genome editing? One is knock-in that will introduce a specific gene. What else?
Sir, I don't have a lot of idea about it. I haven't
Okay.
watched the second week's lectures. I have yet to watch first week's lectures.
Okay. Okay. We'll discuss. You let me know if, based on the discussion, you're able to think about this. One is
Okay, sir.
one is introducing a particular gene that will help in our pathway. Now, let's say I am deleting a gene that is interfering with my pathway. Will that also help as a metabolic engineering approach?
Mm, yes, sir, it can because we can alter a specific formation of product.
Hmm.
We can reduce formation of a undesirable product.
Right.
So,
Yeah. So this is called as knockouts. You are right. So one is knock in, one is knockout. Now, since we are looking at genome editing and not gene editing, there is another set of techniques that can be used, which is altering the expression level of these genes. And to alter the expression level of these genes, we will be looking at the regulatory components in the genome, which would include your promoter and operator region. So one can make changes to the promoter, thereby increasing the amount of enzyme that is being produced. That is also a metabolic engineering approach. I just wanted to ensure that whoever watches this video understands that there is a difference between genome editing and gene editing. So in genome editing, we also consider these changes at the promoter levels or at the operator levels. I hope that was clear. Right. Anyone else? Do you want to try and mention why a particular option is incorrect? Reno Rana or Vetrichelvi? Okay. Does anyone know what mRNA is? Even the Nobel Prize was awarded to a discovery related to mRNA recently. Okay. So miRNA is the short form for microRNA. What this means is this is a small stretch of RNA that is complementary to the precursor for your gene, for your protein. So, because of this small stretch of complementarity, this will be able to bind to either your DNA or your translated transcription product of the gene, which is your RNA. And because it goes and binds to the uh, transcription product, which is your RNA, you will the cell will not be able to produce the gene. Thereby, it will silence the production of the gene. This is also one metabolic engineering strategy. Instead of deleting our gene of interest, we can ensure that the gene is not converting to a protein or a functional enzyme by introducing this miRNA. And the other option is endogenous enzyme overexpression. So this is nothing but increasing the number of copies of enzymes. Again, this is similar to amplification. So more the number of enzyme molecules you have within the cell, more will be your product. So hence this is also a metabolic engineering approach. And the only option that is given here, which is not a metabolic engineering approach, is growing the plants in pots, which is obvious. The next question is, which one of the following is not an advantage for hydrodistillation technique? Option A, very simple, cheap and clean technique. Option B, organic solvent addition is required during distillation. Option C, simplest method for essential oil extraction. And option D, only water is needed as solvent. Again, we can take a couple of minutes, and whoever knows the right answer can raise your hands and start mentioning about the technique here, which is first what is hydrodistillation, and then we can go on to the options. You can take a couple of minutes. Yeah, Renu, do you want to unmute and first mention what hydrodistillation is?
So hybridization is a process in which uh, we add sample in the water to
Okay,
boil it. So that means that we use the polar solvent water as a solvent to
okay.
extract the essential oil.
Can you be a bit louder? It's not very clear.
Uh, in hydrodistillation, we use the water as a solvent to
Hmm.
extract the essential oil.
Okay. Do you know the setup? Like what is connected? Do you heat something? Does the vapor pass through something? Any idea about that?
Uh, it passes through the condenser
Okay.
where the water vapors they get condensed into the uh, water vapors. I mean, vapors they condense into the liquid form.
Hmm. And what is this method used for? Is it used to get an aqueous solution or lipophilic solution? Any idea about that or any comments on that?
It is especially used for the volatile oils.
Okay. So I'll first go through the schematic of how this looks like in hydrodistillation. We'll go through the process and then discard the wrong options. Yeah, you can mute yourself. So this is how the setup looks like. So we take the plant material, we put it here, add water to it, and we heat the mixture till it reaches its boiling point. So once it starts boiling, you have another pipe that is connected here. And what happens is the vapor or the steam that is coming out of the added water that will exit through this. Whereas we also add some cold water inside here. Now the cold water will reduce the temperature, and because of this reduction, whatever came till here, the compounds that came along with the steam, that would start getting slowly condensed and it forms the oil here. And here we utilize the uh, volatile nature of these compounds. And since we only add water and we do not add any organic solvents here, this is an easier method and it is a safe method. Now, going back to the options, if we look at this, we do need water. So that is the right option. And it is a method for oil extraction. That is also the right option. But they also say here organic solvent addition is required during distillation. As we saw here, only water is needed, organic solvent is not needed. So option B is the incorrect option here. Right. The next question is, which among the following methods are used for separation of volatile metabolites? Option A, gas chromatography. Option B, ion exchange chromatography. Option C, affinity chromatography. And option D, high performance liquid chromatography. So we'll take a couple of minutes and then start by discussing what volatile metabolites are. Then we'll start eliminating the wrong options. And once we eliminate the wrong options, we'll also justify why they are wrong and how one of these particular methods is only used for volatile compounds. Okay, so whoever knows the answer can raise your hand. We'll take a couple of minutes and start with the discussion. Yeah, so Nikhil will go first. Nikhil, you'll have to after 2 minutes, you will start with mentioning what is one of the wrong options here, followed by Renu. You will also comment on one of the wrong options here, and then we have schematics. We will look at that as well. Yeah, Nikhil, you can talk about one of the methods here, which is an incorrect option.
Sir, affinity chromatography is incorrect because it is based on uh, immobilization ligand binding with a specific product.
Hmm.
It is very specific in nature,
Hmm.
uh, and it is only work on liquid samples.
Okay, so because you mentioned about liquid samples, do you want to comment on what volatile metabolites mean? We
Uh,
have discussed that earlier today, but yeah, do you want to mention what volatile means here, just so that others can
uh,
are also clear?
Volatile metabolites are those compounds which are in gaseous or vapor phase in room temperature.
Good, good. Yeah. So yeah, like Nikhil mentioned, affinity chromatography is where you have this specificity towards an immobilized uh, acceptor or a receptor. So one specific case that most biologists are familiar with is nickel NTA chromatography, where protein with his tag is bound to agarose beads that contain nickel or nickel nitrile acid, nickel NTA. So that is affinity chromatography. Yeah, Renu. Which option did you want to comment on? An incorrect option?
So ion exchange chromatography and HPLC both are incorrect options because in ion exchange it is the compounds are separated based on their charges,
Hmm.
and HPLC it is a separation of the chemicals in a mixture. We pass the sample through a column, and
Okay.
it is a pressure driven chromatography. So these B, C, D are the incorrect options.
Okay. You did mention about ion exchange chromatography. Do you think pH of the solution matters in ion exchange chromatography?
I don't know that much about in detail about this ion exchange chromatography. I know the basic concept. It is based on the charges of the compound.
Okay. Do you think charge of the compound will change with pH of the solution?
I think so. It will change.
Right. Right. Okay. Huh. Thanks. You can mute. We'll go through the slides. Yeah. So, like they had mentioned, here we have the case of ion exchange chromatograph. Sorry. Here, what is this? This is affinity chromatography, where you have a target analyte and an immobilized receptor. And this is very specific, which is why these are bound. And those that do not have this matching specificity are not bound. And then what we do is once these are bound, we add a competitor, something that can bind to this immobilized receptor with higher binding affinity than this. So those higher binding affinity competitors would compete with these receptor sites and these will elude these. So that is the logic here. Whereas what we have down here is ion exchange chromatography. So here it depends on the net positive charge or net negative charge of the species. I will take one case which is proteins. Proteins have amino acids and amino acids have something called as the R group, the functional group which is actually exposed and not involved in the peptide bond formation. Now, these functional groups can be of any type. It can either be acidic or basic. It can even be phenolic or aliphatic. And because these have these different groups, each of these amino acids that make up a protein will have something called as Zwitter ion. And depending on the pH, this Zwitter ion would change from one particular state to another state. And overall, if you look at it from the protein's point of view, you have something called as Pi, the pH at which the net protein's charge is zero. Now, depending on this pH, we can ensure that a protein can either have a positive charge or a negative charge. When the protein is having a positive charge, it will behave like a cation and it will bind to a negatively charged bead. Whereas if the protein is having a negative charge, it will behave like an anion and it will bind to a positively charged bead. So when we have a solution with our protein of interest and if we want to separate it from other proteins, what we can do is we can simply change the pH of the solution, thereby changing the charge state of the protein. Once we change this, we can use the oppositely charged bead to selectively bind to our protein of interest and then we can later elute them. That is what is given here. And HPLC is a method where, again, you use HPLC for small molecules and for RNA, not for proteins. Some people use it for isolation of peptides, but not for large molecular weight species because the uh, pores are in a way such that they would allow for smaller radius, diameter, or smaller volume molecules. Now in HPLC, again, it's a matter of charge, the overall charge. Um, and here we do apply external pressure, which is why it's called high performance liquid chromatography. So the right option for this uh, part is it is gas chromatography. Now this is the setup for gas chromatography where we have a carrier gas. So the carrier gas is what will carry our volatile molecule, meaning our volatile molecule can dissolve in the gas and it can move. Now, when we put a lot of pressure and put it in one direction, here, what will happen is whatever sample that you inject from here through the injector, and upon applying this carrier gas, your sample will enter this, and this is called as the column. And within the column, it acts in a manner similar to how it works in HPLC, where different molecules of different charges will get eluted at different times. So these are then amplified as signals and we see the chromatogram. The only difference between HPLC and gas chromatography is that since the molecules that we use here are more volatile, they are more soluble in the gaseous state than being in the liquid. So we prefer to use them or separate them and study them using gas chromatography than HPLC. The next question is, name the principal phenolic compound found in root organs of Ananth that is Hemidesmus indicus. And there are four options given here: 2-hydroxy 4-methoxy benzaldehyde, 4-hydroxy 3-methoxy benzaldehyde, epigallocatechin, 4-hydroxy benzoic acid. We can take a couple of minutes and then for this question we can go to the right answer directly because this is just a memory based question. So whoever knows the right answer can take a couple of minutes and then um, answer the question. Okay, so for this question, I was referring to this text. So here they mentioned that the active principle of this fragrance is 2-hydroxy 4-methoxy benzaldehyde, emballed, which is option number A here. The next question is, often microorganisms are used to produce plant specialized metabolites. The process involves several steps. Among the following, which one is not involved in such metabolic engineering? Option A, functional validation of enzymes involved in plant specialized pathways. Option B, gene transfer in the microbe cells. Option C, studying the biosynthetic route in the plant system. And option D, treating the plants with glyphosate. Um, so this is an interesting question. Whoever knows the steps that are involved where one wants to express plant specialized metabolites in microorganisms can raise your hand. We'll start discussing from the first step till we are able to get the metabolite, what all steps are involved. That way, we will know which of these options are a part of the step and which of these is not. And then we'll go to the right answer. Okay. So you can take a couple of minutes, read the question again, think through, and then raise your hand. We'll start discussing the essential steps here. Okay, does anyone want to volunteer? Anyone knows the right answer here? You can unmute.
Sir, answer number D is treating with the glyphosate. It
Hmm.
is an herbicide,
Hmm.
so it is not the right answer.
Okay, so we look at what are the essential steps that we need to do so that we can produce a plant specialized metabolite in microorganism. So the first step that we need to do is first map the enzymes in the pathway so that we know what are the minimum number of enzymes that we need to put into the bacteria or the microbe. Let's assume that the microbe here is bacteria. So the first thing that we need to do is study the biosynthetic route in the plant system. We need to know what is the substrate that is needed, from which pathway is the substrate coming, what are all the transformations that are happening chemically, what is the end product. And because we use enzymes, we also need to be aware of certain things, which is what are all the cofactors that are needed for the enzymes to function so that this plant pathway can be incorporated in microorganism. Now, once we study this, next what we need to do is we need to ensure that we are able to transfer these genes into the microbe cells, because only if the genes are transferred, then the genes will undergo transcription and translation and produce the protein. Now, once we take care of this step, then the protein is produced. Now, one essential thing that we need to consider here is these are plant proteins and plants are eukaryotes. And proteins from eukaryotes are said to undergo post-translational modifications. These could include anything from phosphorylation to the formation of disulfide bridges, anything which we would not have an idea about. So, what one would have to do is when an entire pathway is first being expressed in plants, the first task should be first these proteins should be purified from the microbe and an activity assay of some sort with the correct substrate, correct cofactor should be done to see if the product is being formed. This would mean that the enzyme can be produced within the bacteria and the enzyme is functional. This way, we will ensure that whatever minimal is required for the enzyme to be active within the microbial environment is taken care of. So now we know that the pathway is active. So these are the minimal steps that one needs to do when a plant pathway is being incorporated in a microbe. And like Nikhil mentioned, treating plants with glyphosate is irrelevant for this metabolic engineering because it's a herbicide and it would only affect the weeds. The next question is, decaffeinated coffee can be produced by genetic engineering using one of the following strategies. Sorry. Identify the correct one. And they give us four options. Reducing theobromine and caffeine content by silencing theobromine synthase gene. Increasing theobromine and reducing caffeine content by overexpressing theobromine synthase gene. Increasing theobromine and reducing caffeine content by overexpressing caffeine synthase gene. Decreasing theobromine and increasing caffeine content by overexpressing caffeine synthase gene. So, in order to understand how to answer this question, we need to be aware of the pathway. So, you can take a couple of minutes and maybe check the internet also if you have, uh, or check your notes to see how caffeine is being produced. Which of these is being produced? Either theobromine is the precursor, or is caffeine the precursor, or if both of these are a product of the same gene. You can check that, and that will help us answer this question. You can take a couple of minutes, then we look at the pathway that produces these compounds. Okay. Okay, does anyone know the pathway for coffee production? Here it's the pathway to produce theobromine and caffeine. You can unmute if you know the pathway. Okay, so this is how the pathway looks like. So we have something called as xanthosine. This is a part of your nucleic acid biosynthesis pathway. And you have SAM and SAH. SAM is acetyl methionine, whereas SAH is a reduced product of SAM. Now, this is involved in methylation. So, SAM and SAH, uh, these would tell you that these are the necessary cofactors that are involved in methylation. If you see the difference between xanthosine and 7-methyl xanthosine, you have an addition of methyl group at this nitrogen. So, and in this pathway, after 7-methyl xanthosine is produced, or xanthosine is produced, it gets uh, the ribose is removed. You get 7-methyl xanthine, and then it is again methylated to get theobromine, and then this is again methylated to get caffeine. So, the question is, how would you produce decaffeinated coffee? Which means that how would you reduce the production of caffeine? Which means you have to either stop this step or stop this step. If you produce theobromine and stop step number 4, which is the caffeine synthase, you can reduce the production of caffeine. Or you can reduce the production of theobromine itself. That would also reduce the production of caffeine. And if we look at the options here, they say reducing theobromine and caffeine content by silencing theobromine synthase gene. Because theobromine is the one that gives caffeine, if you reduce theobromine production itself, that will help. And this is the right option here. Whereas if you look at other options, which is increase theobromine and reduce caffeine, this will not be possible because if you overexpress theobromine synthase, it will give you more of theobromine. And more of theobromine means as long as you don't affect the other gene, caffeine synthase, you will have more of caffeine. And if you overexpress caffeine synthase, it will produce more of caffeine and it will not reduce caffeine. And that's why option A is correct here. The next question is tDNA of naturally occurring TI plasmids contain genes for phytohormone synthesis, which will cause tumor after infection in host plant. The concerned phytohormones are option A, auxin and gibberellin, option B, cytokinin and abscisic acid, option C, abscisic acid and cytokinin, and option D, auxin and cytokinin. So, whoever knows the answer for this question, you can first explain what tDNA is, what TI plasmid is, and then we'll take it from there. We also have a few schematics to explain what these are. So, finally, we'll go to the options here. Okay. Okay, so tDNA is a part of TA plasmid. So this TA plasmid has something called as origin of replication, virulence region, and it has a tDNA region. Other than this, it also has this open catabolism. Now within this tDNA, it has been segmented into left border, right border, within which you have a few uh, coding region, which is the which codes for your auxin, cytokinin, and opine. Now, this is naturally present in Agrobacterium tumefaciens, and this tDNA is the place where people who want to insert a particular gene or introduce a particular gene in plants introduce their gene of interest, because this is not very essential. The so once they insert it, they can introduce it into the plant cells in culture, and the tDNA carrying the new gene, it will start expressing with that particular gene of interest. If you put a GFP, which is a green fluorescent protein, you might be able to see the cells glowing. So coming back to the question, they asked what are the concerned phytohormones that are present in tDNA. So that is auxin and cytokinin, as shown here. The next question is match the chromatography technique with their preferable detectors. And it gives two columns. Column one is with HPLC and gas chromatography, and in column two we have photodiode array, mass spectrometer, flame ionization detector, refractive index detector, electroantenographic detector, thermal conductivity detector, and evaporative light scattering detector. There is an easy way to rule out the wrong options here. I would like to give you a hint for this question. It is a detector that you can only use with gas chromatography and not HPLC. That is present here. If you know that option, you will be able to get the right option in the first choice itself. So you can think about what is specific in gas chromatography and how detection is done there. Although we'll be going through that here. We have the schematics for each of these. So uh, you can take a couple of minutes and think about what is so specific about gas chromatography that it requires that the detector that is used in gas chromatography, this specific one detector, cannot be used in HPLC. You can think about that and then raise your hands. Okay, so I was mentioning about a particular kind of detector that is only, yeah, who is it? Yeah, Renu Rama. Okay, Renu. I was giving a hint to identify the right option here. Um, I mentioned that one of these detectors can only be applied with gas chromatography. We're able to find what the detector is. Yeah, Renu Rana, you can unmute.
Flame ionization directed detector.
Yeah, do you know why it is only used for gas chromatography?
Because gas chromatography is used for the volatile components.
Huh. And how does flame ionization detector work?
I don't know the working of this detector, but uh, it detects. I think it detects the volatile compounds.
Hmm.
Uh, that's why we can use it in the gas chromatography.
Yeah, volatile compounds, that is one, but people have there is a possibility that you can use volatile compounds with HPLC as well. The thing that you need to know about gas chromatography is in gas chromatography, your compound is not a liquid anymore. So even though it is volatile, it is not a liquid anymore. So this is how your flame ionization detector looks like. So earlier we saw what gas chromatography chamber looks like. So you inject it, a gas carries your injected sample, it goes through the column, and depending on the affinity, some of it will elute first, some of it will elute later. Now, in this detector part, we have something like this, the flame ionization detector. So what will happen is, you would again add some gas here and the air here, and there will be a flame here that will be ignited. So when your volatile compound comes here, that would alter this, the readout here, which will what be amplified. So because these are gaseous, and because these come as ions, they can, upon hitting this collector, produce a particular current, which is what is later on amplified. So this is only possible if the molecules are gas. If it's a liquid, this will not be able to work and hit the detector to produce the current. Which is why for only for gas chromatography we can use flame ionization detector. We can also look at other detectors here. For example, the photodiode detector. So here what happens is we have a lamp that will emit light, and the light will pass through something called as the flow cell here. I'll zoom this a bit. So it will pass through something called as the flow cell here, and we have the diffraction grating as well. So when the light passes on the photodiode, which is on an IC chip, the photodiode will convert the light into electrical signal. Now, the liquid will also pass through the flow cell, which will uh, interfere with the amount of light that is falling onto your photodiode. That will tell you how much of interference has happened or how much of absorbance has happened. This is the principle for photodiode array detector. So here, this we can use it for both proteins and for small molecules that can absorb at a specific wavelength. Then we have refractive index detector. Here we measure one application that I am aware of is called as um, SECMALS, where we are able to get an idea about the shape of the protein by passing it through a column and the scattering of the protein. So what happens in so this refractive index detector is used in secmols. In secmols, you pass a protein, you get an idea about the molecular weight, and when the protein is passing through the reference cell, because it is the protein solution is very different from the normal blank solution or the buffer solution, there will be a difference in the refractive index. Now, refractive index tells you if the light, how much of the light can pass through and at what angle it is bent, depending on the nature of the solution and the number of molecules or the nature of the molecules in the solution. So, based on this, people were able to estimate the shape or the size profile of protein molecules in solution in sigmoids. Other than refractive index, we also have uh, electroantenographic detector. This is not relevant to detection of small molecules, but rather this is used for uh, connecting with live insects, insects that are alive. What people do is they use these detectors to connect with the antenna, the those stick-like objects that are present on insects, and try to identify what molecules are produced there. Whereas thermal conductivity detector compares thermal conductivity of the carrier gas to that of the sample. Again, this can be used in gas chromatography majorly. So here your carrier gas will be the gas which you pump in, which will carry your solute of interest. And it uses a heated metal filament to sense the changes in the thermal conductivity of the gas. So, in the presence and absence of molecules, the ability to transfer heat from one position to another position would vary. Based on this principle, this detector works. And evaporative light scattering, that is another method where the detector can detect the compound based on light that is scattered by small molecules or small particles that are formed when the mobile phase of the sample evaporates. So this would allow for detection of non-UV absorbing compounds like carbohydrates, lipids, polymers, which are often used in conjunction with HPLC analysis. So we know generally that compounds like proteins absorb at 280 nanometers, or some molecules absorb at 254 to 300 nanometers. There could be other lipids or carbohydrates or polymers that do not absorb at this range. So for those cases, we use evaporative light scattering. And finally, the last question for today's problem solving session is that among the following options, which one is not a part of wild type Agrobacterium tumefaciens plasmid? The options are cytokinin biosynthesis gene, multiple cloning site, virulence genes, and opine catabolism genes. So, to understand this question, we need to look at the TI plasmid. You can take a couple of minutes, think about this question, and whoever knows the answer can unmute. Okay, so this TA plasmid, it's a plasmid that is naturally occurring in uh, Agrobacterium tumefaciens. Now, the purpose of this TA plasmid in Agrobacterium tumefaciens was to infect these. So it did not have an MCS. MCS is multiple cloning site, meaning it will have multiple, the sequence is designed in such a way you will have restriction digestion sites. This is engineered in plasmids. It is not naturally present. So, although it contains this open catabolism, virulence region, and this phytohormone producing genes, it doesn't have this MCS. In order for our purpose to introduce genes into plants, this TA plasmid is modified to have MCS. So, this multiple cloning site is the wrong option for this question. So, with this, we come to an end for today's problem solving sessions. If you have any doubts, you can ask me. I'll be staying on the call. Otherwise, you can the class is over today.
Thank you, sir, for the doubt clearing class. I'll be watching week's second lectures to
Sure,
clarify my basis
sure,
of this lecture.
sure. You can also join next week's class if you have any doubts after watching the videos. Thanks for joining.
Okay, sir. Thank you, sir.
Garima, since you've joined the class now, so we have discussed all the questions for this week's lectures. I'll be sharing this PPT today. Um, if you have any specific questions, you can ask me. Or if you want me to go through the questions again, you can let me know. I can go through that for you.